Hello everyone, welcome to PathoMed. Today let us discuss about an important topic from the chapter of Neoplasia, that is Microbial Carcinogenesis. A brief introduction is the infecton, that is the microorganism causing infection. It triggers cell proliferation, which is initially polyclonal and later it becomes monoclonal. Polyclonal means different cells have different genetic compositions. The rapidly dividing cells individually they have different genomic compositions. Later, upon acquiring driver mutations, they become monoclonal. Monoclonal means all the cells have same identical genetic composition. Driver mutations they change the DNA sequences in the cells, making them like proto-oncogenic and later ultimately causing cancer. It becomes a tumor. So polyclonal cells become monoclonal because of acquiring driver mutations in rapidly dividing cells. So let us study about few viruses and bacteria that are oncogenic. Under bacteria, under viruses, first is RNA and DNA viruses. Under RNA viruses, we have human T-cell leukemia virus 1 and also hepatitis C virus. Hepatitis C virus, which is a single-stranded RNA virus, but hepatitis B is a DNA virus. Since hepatitis C and B, both of these viruses have similar mechanism and known to cause similar cancers, we'll study them together under one roof. So let us first start with HTLV1. Human T-cell leukemia virus is well known to cause adult T-cell leukemia lymphoma, that is ATLL. HTLV1 causes ATLL. And this virus has a tropism, that is affinity for CD4 plus T cells. This indicates that CD4 plus T cells they become the major target for this virus, making them neoplastic. The modes of transmission of this virus are sexual mode, blood products, that is like blood transfusions, and breastfeeding. The site of viral integration in host chromosome is random. What does this mean? This means different cancers. I mean, there are many types of cancers. Different types of cancers have different. In those cells, the viral integration into the host genome is different. If suppose a given there's a number one cancer, for example, cancer X is there. In that cancer X, the tumor cells, the integration of viral genome into the host genome is at a particular location. In cancer Y, this site it is different. In cancer Z, the viral integration, the site of viral integration is different. This is said to be random viral integration. But for a given particular cancer, for example, cancer X, for a given particular cancer, all the tumor cells, the site of integration of the viral genome into the host cell genome is same, identical. That is said to be clonal. So it is a clonal pattern. This HTLV1 genome, it has certain components to remember: GAG, POL, ENV, and of particular significance are the TAX and HBZ. TAX is essential for viral replication. HBZ is a transcription factor. Transcription factor means it receives the signals, transmits it to the nucleus. It interferes with the genome expression. This tax and HPZ on the whole, these have effect on intracellular signals. They regulate, that is something to do with cell survival, cell growth and survival. And they induce genomic instability. They inhibit senescence. Senescence means to remain quiet, to remain silent. So if you inhibit senescence, the cell does not remain silent. It becomes aggressive. It multiplies very rapidly. The cells run amok. Now we'll discuss about the DNA viruses. Among it, the first one, human papilloma virus. We are well known with the low risk and the high risk variants. The low risk variant are the 6 and 11. Low numbers, I mean small numbers 6 and 11, low risk. They are known to cause the benign warts, benign genital warts. Why are they benign? Because the low risk variants The viral genome is maintained in a non-integrated episomal form. For example, this is the host cell, this is the host DNA, this is the host DNA. Genetic material is present within the nucleus of the host cell. When it is infected with this virus, HPV virus, the HPV virus, it enters the nucleus, but it remains like non-integrated, does not integrate, does not mix, does not attach to host chromosome. It remains as a separate entity, and they replicate, both the host and the viral genome, they replicate separately. So it is said to be non-integrated, it is said to be non-integrated episomal form. So the reason why it is of benign nature. Whereas the high risk variants like 16, 18, higher numbers are high risk. They are well known to cause squamous cell carcinoma of cervix, adenocarcinoma of cervix. These are well known cancers. Why it is cancerous? Because the genome is integrated in the host genome. This is host cell, host DNA containing the genetic material, and the viral genome becomes a part of the host genome and it replicates along with it. There's interference, it interferes, it mixes with the host genome. That's the reason why it is integrated. That's the reason why it is more cancerous. And like HTLV1, HPV also, the viral integration is random. For random means adenocarcinoma, squamous cell carcinoma. These two different cancers have different sites for viral integration, but the pattern is clonal. Means for squamous cell carcinoma, all the cells have same site for viral integration. For adenocarcinoma, all the cells have same site for viral integration. That is said to be clonal pattern. And this viral integration, it disrupts E1 and E2 open reading frames. E1, E2 open reading frames. This leads to loss of E2. This leads to loss of E2 viral repressor. Hence, there's increased expression of E6, E7 genes expressions. So these E6 and E7, they have particular significance. E6, they have two functions. First, it binds to P53. P53 is the guardian of the genome, policeman of the genome. It is a tumor suppressor gene. It is a tumor suppressor gene. It is a tumor suppressor gene. This E6, what does this E6 do? It comes and binds to this tumor suppressor gene. It degrades it. It degrades tumor suppressor gene. When tumor suppressor gene is lost, the cells, they grow, they survive, they proliferate uncontrollably. And also, the second function is they stimulate TERT, that is telomerase reverse transcriptase enzyme. So, because of this increased telomerase adhesions of chromosomes, this results in increased tyrosine kinase activity. Increased tyrosine kinases means increased cell signaling pathway, increase in cell cycle progression, increased cell growth. So, by these two mechanisms, they make the cell immortal. That is, the damaged cell did not undergo apoptosis. They remain, they survive, they remain, they proliferate, they make progenies, they make colonies. The cell becomes immortal, they do not die. And interestingly, the E6 from the high risk variant, high risk HPV, it has a higher affinity for P53 than that E6 derived from the low risk variant. Low risk has low affinity, high risk has high affinity. Hence, this explains the difference in cancer risk. Higher risk variants have high cancer risk because higher risk variants have high affinity for P53. And E7, it has these two functions. Firstly, it speeds the cell cycle through G1S checkpoint. G1S checkpoint. That means the damaged cells, they cross over, they easily snap off through, or they easily escape the checkpoint. They easily escape of this checkpoint and enter the cell cycle. It speeds up the G1S checkpoint. And also the E7, it binds to RB protein, which is a governor of the genome. RB protein displaces E2F transcription factor, E2F transcription factor, and hence it in this way it increases the cell cycle progression. And another, it inactivates the inhibitors. Cyclin-dependent kinase inhibitors are inactivated. P21, P27 cyclin-dependent kinase inhibitors are inactivated. Inactivation means the cell cycle progression occurs. Similarly, same like E6, the E7 also, the high risk variant has high affinity for RB and they bind and activate cyclins. Cyclins are activated means they are helpful for cell cycle progression, the cell cycle progress, they run G1, MS, like that, they run in the cell cycle. And this high risk HPV, they express certain oncogenic proteins that have following functions. They inactivate tumor suppressor genes. They inactivate the tumor suppressor genes. They activate cyclins. They activate cyclins. Tumor suppressor genes are inactivated means uncontrolled growth. Cyclin activation means they are helpful for cell cycle. They activate the cell cycle. They inhibit apoptosis. They inhibit apoptosis means the cells, damaged cells do not die. They remain as such, they survive. And they combat cellular senescence. They combat cellular senescence. They are resistant to cellular senescence. Cells do not remain silent. They become aggressive. The HPV viral infection as such is not carcinogenic. One-time infection with HPV, the woman does not develop cancer. They are repeated insults to it. The HPV infection, along with certain other insults in concert with other environmental factors like cigarette smoking, dietary deficiencies, hormonal changes, coexisting microbial infections. All this together, they play a role in pathogenesis of cervical cancer. Coming to Epstein-Barr virus, the most common Epstein-Barr virus associated tumors are lymphomas derived from B cell, that is B cell lymphoma, and nasopharyngeal carcinomas. Also, they are also known to cause rare type of sarcomas as well. Firstly, let us know the structure, the function, the functionality of the virus. This EBV, this is the Epstein-Barr virus, which I have drawn in gray color. This Epstein-Barr virus, it has a surface glycoprotein. This is the surface glycoprotein. Why is it helpful? The function of the surface glycoprotein, it recognizes the B cell receptor. This is the B cell. B cell. It has receptors on its surface, which is a complement receptor. CD21, which is a complement receptor. CD21 is a complement receptor. This GP, glycoprotein, it recognizes this receptor, binds to it, and infects the B cells. Normally, in a normal, healthy individual, the B cell, infected B cells, they are removed by the T cells. The cytotoxic T cells recognize this EBV and B cell complex and are killed. They kill it, and hence the B cell growth is kept under check. And the infection, the latent infection of B cells with EBV is said to be latent. Latency means there's no viral replication and no killing of B cells. Typically, the Epstein-Barr virus, the genome has three components: LMP1, that is latent membrane protein 1, EBNA2, Epstein-Barr virus nuclear antigen 2, VIL10, viral interleukin 10. There are certain functions to each of it. First, coming to latent membrane protein 1, LMP1, they activate signaling pathways. Few of the pathways are NFKB, JAK-STAT, sorry, JAK-STAT, JAK-STAT, and also they activate BCL2. BCL2 is anti-apoptotic factor. That means the damaged cells, they do not undergo apoptosis. They survive, they proliferate. And EBNA2, they activate cyclin D. Cyclin D necessary for cell cycle. Cell cycle. And they also activate SRC family of proto-oncogenes. This family SRC is a family of tyrosine kinases which are proto-oncogenes. Ultimately, they are involved in tumor. And viral interleukin 10. Interleukin 10, this is anti-inflammatory kind of thing. They suppress the T cell activation. They suppress T cell activation. T cells are not activated. Firstly, coming to the pathogenesis in Burkitt lymphoma, I mean the EBV known to cause Burkitt lymphoma, the Epstein-Barr virus, they do not express these proteins as normally they express this LMP1, EBNA2. But in Burkitt lymphoma, these are not expressed. These are not expressed. That means they are not directly oncogenic. This Epstein-Barr virus, instead, it acts as a polyclonal B cell mitogen, like it causes translocation. By causing a translocation of chromosomes 8 and 14, it activates c-MYC. It activates the c-MYC. C-MYC is a proto-oncogene, which ultimately leads into full-blown cancer. Although they do not express the EBV proteins LMP1, EBNA2, but by causing a translocation, they activate c-MYC and ultimately causing full-blown cancer. In immunocompromised patients like AIDS, and those patients on immunosuppressants. For example, patients receiving immunosuppressants to prevent graft versus host rejection. For those such patients, they lack T cells. They lack T cells. They lack T cells. So the Epstein-Barr virus in them is directly oncogenic. That means directly oncogenic means the true nature. They express LMP1 and EBNA2, the functions we have seen earlier to cause cell cycle progression. In nasopharyngeal carcinoma caused by EBV, the Epstein-Barr virus, they express LMP1. LMP1, we have known that it activates certain pathways. That is, here they activate NF-kB pathway, cell signaling pathway. They upregulate the expression of vascular endothelial growth factor, fibroblast growth factor, matrix metalloproteinase 9, MMP9, COX-2. All these factors, they are required for cell survival, spread, proliferation. Cell survival, spread as well, metastasize, to invade and metastasize. They ultimately cause oncogenesis. Now coming to hepatitis B and C. Hepatitis B is a DNA virus, C is a RNA virus. We'll study them together. 70 to 85% of hepatocellular carcinomas, they are associated with viral infection with HBV and HCV. The oncogenicity of this viral infections, they are multifactorial. One such theory, firstly, is because of that infection, there's a chronic inflammation within the liver. Chronic inflammation due to infection, because of which the hepatocytes die. There's hepatocyte death. To compensate the death, to compensate the loss of hepatocytes, there will be regenerative process. So there's hepatocyte proliferation. The hepatocytes grow in an uncontrolled way. With time, they incur genetic damage. This genetically damaged cells, without any inhibitions, they grow. They ultimately lead to HCC. Other mechanism is activated because of this infection and inflammation. There is activated immune cells. The activated immune cells during the chronic inflammation, they produce growth factors, growth factors, cytokines, chemokines, chemokines, which are the mediators of inflammation, and certain bioactive substances. Most importantly is the reactive oxygen species. Reactive oxygen species is a genotoxic and mutagenic. They damage the genome. And other pathway is the NF-kB pathway, cell signaling pathway activated and prevent apoptosis. The cell survives, that is damaged cell survives. The dividing hepatocytes, they incur genotoxic stress. They incur genotoxic stress. They accumulate mutations over time and ultimately lead to tumor. And other mechanism is the HBV genome. The HBV genome, it carries a component HBX component. This activates a transcription factor. Transcription factor reaches the nucleus, activates cell signaling pathways. This viral integration of this, the HPV genome viral integration also causes certain disturbances in the host chromosome structure. Host chromosome structure destroyed, which dysregulate oncogenes and tumor suppressor genes. When these are dysregulated, tumor grows. And HCV has a core protein, HCV core protein directly a direct effect on tumorogenicity. Coming to this bacteria, H. pylori, Helicobacter pylori. It is the first ever bacterium to be classified as a carcinogen. These are associated with gastric adenocarcinoma and gastric lymphomas. The mechanism of this are the H. pylori it expresses a gene, the CAG-A gene which penetrate the gastric epithelial cells and they initiate the cascade, signaling cascade leading to uncontrolled growth. And also they stimulate reactive T cells. The reactive T cells synthesize cytokines which in turn stimulate B cell. So there is a B cell proliferation. There is a B cell proliferation. T cells stimulate B cells, which ultimately become monoclonal maltoma. The sequential changes in the gastric mucosa because of this H. pylori infection are there is firstly inflammation. Infection leads to inflammation, gastritis. Chronic gastritis leads to atrophy of the epithelium. Because of atrophy, I mean, this again later this metaplasia. One epithelium changes into the other type, metaplasia. And in compensation, is proliferation, sustained epithelial proliferation. Sustained because of rapidly dividing upon time, they acquire mutations, accumulate genomic mutations, ultimately causing gastric carcinomas. Yes. With this, we have completed the microbial carcinogenesis. Thank you all and have a nice day.